Hi, my name's Elisa and I'm going to be talking about my project, which is called Harnessing the eDNA in Tidal Flows to Assess Marine Biodiversity. Some background on my project first. Environmental DNA, or eDNA, is a term used to describe genetic material left in the environment by organisms, such as skin cells, metabolic waste, gametes, etc. It can be collected from soil, sediment, or water. So, for example, you can take a sample of water, amplify the DNA present, and ultimately figure out what species were present without ever having to see them. It has previously been used to track rare or invasive species and has also been used in biomonitoring. However, more work has been done on freshwater systems than in marine. For example, there have been many studies published about transport distance in rivers. Unfortunately, this process is harder to quantify in marine systems, both due to larger volumes of water, which could dilute eDNA, and also due to the non-linear nature of the marine environment. The effect of currents and tides on the fate of eDNA is often not properly integrated, therefore it is difficult to interpret results on a spatial scale. In order to be able to use these results for management of marine environments, it is critical to know if the results show a local signal of eDNA or if it has been transported from elsewhere. Unfortunately, I don't really have time to talk about my study area of Strangford Lough, which is a sea inlet here in Northern Ireland, so instead I've included some drone footage of it. In order to investigate some of the transport dynamics of eDNA, we will use a point source of non-native eDNA. To do this, we will use an outflow pipe of the Explorer's aquarium, which flows into the loch. This source is ideal as Explorer's has a variety of native and non-native marine species present, so that if any non-native DNA is found in the sequence data, we can be sure of its origin. Samples will be collected in triplicate along a transect running in two opposite directions. We will be using two methods to get results: a species-specific approach which involves qPCR, and another which is used for community composition called metabarcoding. The results of these approaches will be compared to test sensitivity. Due to imperfect detection of species, the idea is to use a species occupancy model to determine the probability of detection or non-detection of a chosen species with increasing distance from the source. Results will also be compared to a pre-existing hydrodynamic model of Strangford Lough to see if they correlate with water movement. Going forward, I'm also hoping to investigate tidal transport dynamics and spatial distribution of eDNA, the impact of salinity and water turbidity on eDNA detection, and invasive species within the loch, potentially developing a qPCR assay for one of these species. So thank you very much for listening to my talk, and I'll leave you with my question for the Quadrat quiz.